cancer at the moment. So we'll be presenting on the topic Biomass and Nanoparticles for Cancer Detection and Treatment, where biomass basically means bioelectromechanical systems. Now, I'll just basically try to give an overview of what cancer is and what we are up to. Cancer is basically cells that have actually refused to obey, most especially the genes that are supposed to fail to do so. So they are growing abnormally, different from the normal division of cells. Now, within the period of 2011, World Health Organization estimated 84 million deaths, all arising from cancer, representing 22 deaths on all diseases. And the statistics across a part of the world. It has been shown from the graph we have there, most especially the one up there, that cancer alone kills more than when we consider tuberculosis, HIV, and malaria, which means that there is a lot we have to do. Is early detection and treatment, and most of the treatment methods we have these days also have their own chemotherapy. It basically releases toxic chemical with the hope of only 1% of this drug getting to our cells. That 99% of drug concentration, what does it do to the normal cells? It means that it's basically going to put the patient under different conditions. Other challenges may be the spread of disease cells, most especially with surgery. After they try to cut as much as possible around the tumor they remove, but in that process, the blood flow tends to carry cancer cells to receiving radiotherapy. It's not advisable at all, and that is one of the fear of Japan and the rest, because of their spread cancer to all other parts of the body, and that treatment is. When we look at this graph, it tells us this is the trend for developed countries, but that is for developing countries. Africa and Asia seem to be facing that challenge. And what are we doing on our own part of this? The thing we are suggesting is, can we use nanotherapy, or can we use bioelectromechanical systems that will really target the disease at the specific site of interest, since all the other methods like that. In our approach, we are using temperature-sensitive polymers in the form of gels that are capable of carrying cancer drugs, such that when we trigger their temperature a little wide, they have a lower critical solution temperature, they respond to the change in temperature and then deliver the drug directly to the area. We also try as much as possible to control the concentration of our delivery so that we will not go to the toxic region, or the concentration will not be less, but rather within the therapeutic window, which is the acceptable dose for. Now, over the years, this is the effort that we have been able to achieve. We're able to carry out this research, and through the collaborative effort of Shedder Science and Technology, we're able to produce a local drug, what we call a prodigocin, out of a bacteria called Serratia marcensis from the soil ground of Shedder Science. So from that, we're able to culture cancer cells, and we basically induce that to see the effect of cell survival in terms of drug concentration. And then the standard drug we used was also paclitaxel. And up there is just to show one of the work done by our previous student where they used paclitaxel to shrink the tumor, which shows that when the concentration increases, the cancer cells, the tumor reduces. But when the drug is getting used up from the device, then the tumor begins to grow again. So we need to really concentrate and control what amount of drug is capable, and then how the device can deliver the drug for us. So in summary, this is how the drug delivery is going to look like in the future. We hope to have a sensor that will control the drug for us to be able to get the target concentration, then for it to stop when we notice that, trying to see how we can incorporate an identification tag using the radio frequency, and the work is also being carried out by Kobna Kandapa, one of my colleagues. Another thing is, this device is biocompatible, but we need to know how its interaction with the biological system is going to be. It's also looking at the interaction of the biological system with the drug, with the device, and in a way, he's trying to texture the surface of the substrate such that after the drug is being delivered, then this device is going to home cells so that it will form part of the body. And that is what we are looking at. So in the future, we'll be having a multimodal device that will be able to perform all these functions. And we are looking to the near future to be able to carry out the animal work, and most especially also the human trials.
From the Department of Material Science and Engineering, working on the biosynthesis of gold nanoparticles using bacteria and plant extracts. What are nanoparticles? Nanoparticles, they are particles within the range of 1 to 100 nanometers. Because in nanoscience, the size is very important. Now, how do we produce nanoparticles? For gold nanoparticles, you can do it chemically or biologically. Now, for chemical synthesis, it's not advised because it's been found to be toxic. They are not biochemicals we use in the synthesis that are very expensive. As compared to the biological synthesis, the bacteria we've used, we got them from our labs here in Nigeria, and then the plants, especially the plants you're seeing here, nuclear acidifolia, we've been able to use it to produce gold nanoparticles within a matter of, unlike the chemically synthesized nanoparticles that will take hours to produce. Now, the pictures, shapes, and sizes are very important. Biosynthesis gives us an advantage of being able to control the shapes and sizes of the nanoparticles you produce as against the chemical synthesized one. The down here is what we're seeing here is the nanoparticles produced from Serratia marcescens at the pH of 4. You'll be getting these different shapes of the nanoparticles. Whereas for chemical synthesis, it will just give you spherical. In our approach, what is the essence of these nanoparticles? Our interest is in using them for cancer detection and therapy. And like my colleague pointed out, the major problem of cancer in Africa is that of early detection. That is why the mortality rate is high. In essence, if you compare the cancer level in developed countries, as in the incidence rate in developed countries is very high, but the mortality rate in Africa is on the increase of a problem of early detection. Therefore, when we use nanoparticles to label the cancer cells, it will aid in the early detection, it will aid in the vision. For instance, the picture we have here, these are magnetic nanoparticles. The work that is done by my colleague, John David Obayemi, he produces magnetized nanoparticles. If you submit the patient to a magnetic resonance imaging, it will generate magnetic fields whereby the cancer cells, the vision will be very clear. That's what you can see there. And then whereas if you don't, the vision will see without nanoparticles will be very blurred. It will be difficult for you to confirm is this really cancer or not. Again, the down part is where we label the cancer cells with gold nanoparticles. Actually, this one will give you a better and very clearer vision of the cancerous cells. This shows basically because the essence of this work is that the nanoparticles, they are specific. They will really attach to cancerous cells. You can see the dark areas, those are the areas, the cells that are normal cells, whereas the nanoparticles only label cancerous cells. So that when you view it, you'll be able to see the extent of the malignancy of the cancer. You see the extent where it has spread, and be able to detect it and producing the nanoparticles. We'll go ahead to functionalize them. We don't just use them. There are some receptors that are overexpressed on breast cancer cells. We have the folate receptors, we have the LHRH receptors. So the essence is you functionalize, you conjugate the nanoparticles with these receptors. You can even use some antigens. Like my colleague, again, he used LHRH to functionalize his magnetized nanoparticle, whereas for the acid to functionalize them. Then when you inject, again, we encapsulate the anti-cancer drug. The drug we're working with is prodigiocin. Like he said, we discovered that we produce it in Shesco, in our labs there in Shesco. So when we introduce the drug into the animal, this picture shows us what goes on within the cells. Firstly, we see the conjugated nanoparticles containing the drugs. When they get to the cancerous cells, they will attach specifically to the sites, to the receptors. The part of the cell that doesn't contain the receptors, the nanoparticles will not attach to them. They are really specific, the nanoparticles. These are an advantage of using nanoparticles to target cancer as against chemotherapy, because this one will actually go straight to the cancerous cells. Now, while inside the cytoplasm of the cell, then it will release the drug. Thereby, it will now kill the cancer. We're still going to use the nanoparticles for what is called hyperthermia, that is increase in the body temperature. For the gold nanoparticles, it has the ability of absorbing light. When you excite it with a laser beam, to absorb energy, which is to translate into heat energy that will now cause local heating of the cells and thereby killing the cells. The same thing can be done with magnetite nanoparticles. If you introduce your nanoparticles into either a lab animal or a human being, you put the person in magnetic resonance imaging system, then to generate magnetic field that will cause local heating of the cells and thereby also shrinking the future of our work is we are looking at why we can use both hyperthermia and targeted drug delivery in the treatment of cancer. That is where we are going, whereby we combine the effects of hyperthermia and targeted drug delivery. Thank you.